We are jumping straight into the microscopic warfare of the phylum Apicomplexa today. We're going to break down the exact molecular tactics and structural mechanisms that Toxoplasma, Plasmodium, and intestinal coccidia use to completely hijack our cells. Here's the roadmap for this explainer. 1. Taxonomy of phylum Apicomplexa. 2. Morphology of the apical complex. 3. Cellular invasion and the glidosome. 4. Morphology of Toxoplasma gondii. 5. Morphology of Plasmodium species. And 6. Morphology of intestinal coccidia. Section 1. Taxonomy of phylum Apicomplexa. Let's establish exactly what we're dealing with here. Apicomplexans are classified within the supergroup Chromalveolata, and they are obligate intracellular parasites. That word obligate is absolutely crucial. It means their entire existence, their survival, completely hinges on successfully breaking into a host cell. And their master key for breaching that cellular wall? It's a highly specialized structure called the apical complex. Section 2. Morphology of the apical complex. Let's zoom in and look at this primary weapon up close. Looking at the anatomical layout, right at the very tip, you've got the conoid. It's this tightly coiled network of microtubules. Think of it almost like a microscopic drill bit. It gives the parasite the strict physical rigidity it needs to withstand the massive mechanical stress of pushing into a host membrane. Right behind that drill bit are the micronemes and rhopteres. These are essentially the payload bays, loaded with the biochemical tools required to actually execute the invasion. So, how does this attack sequence play out? It's incredibly fast. First, those micronemes release adhesins, specifically proteins like MIC2. This anchors the parasite to the host cell. Once it's tethered, the rhopteres step in, injecting RON and ROP proteins directly into the host membrane to create what's called a moving junction. Now, notice the apoclast here. This is an evolutionary leftover from an ancient cyanobacterial ancestor. Its job is to synthesize vital lipids, specifically isopentenyl pyrophosphate, or IPP, which the parasite absolutely needs to survive. But because it uses ancient prokaryotic machinery to do this, it creates a massive Achilles heel. It's the exact reason we can effectively treat these complex eukaryotic invaders with prokaryotic targeting antibiotics like clindamycin. Section 3. Cellular invasion and the glidosome. How do these organelles actually force their way inside? Keep in mind, this isn't some passive diffusion. This is forced entry. After that initial binding, the parasite's rhopteres inject the RON2 protein directly into the host cell's membrane. Then, the parasite secretes AMA1 from its micronemes, which locks right onto that newly embedded RON2. It's a brilliant lock and key mechanism that effectively staples the two cells together, finalizing that moving junction. Finally, the parasite secretes toxophilin. This is an enzyme that actively dissolves the host cell's internal actin skeleton. By destroying that structural resistance, the parasite can just rev its internal motor, the glidosome, and push itself straight inside, all without ever popping the host cell membrane. Section 4. Morphology of Toxoplasma gondii. Let's see how these molecular concepts translate to physical structures. In host tissue, Toxoplasma gondii takes 3 distinct visual forms. First, you have the tachyzoites. You can easily recognize them by their crescent or arc shape, usually measuring about 5 to 8 micrometers. When these rapidly divide inside a single host cell, they pack tightly together to form a pseudocyst. Eventually, when the host's immune system starts putting the pressure on, the infection progresses into its chronic phase. This forms a true tissue cyst packed with slow-growing bradyzoates that can hide out indefinitely in brain or muscle tissue. This physical shift from a tachyzoite to a bradyzoite requires a massive metabolic rewiring, and it directly dictates the clinical pathology. Tachyzoites are your acute attackers. They divide rapidly, rely on aerobic metabolism, and express the SAG1 surface protein for quick cellular invasion. But when the immune system turns up the heat, the parasite has to pivot to survive. It transforms into a bradyzoite, swapping its surface protein expression from SAG1 over to SAG4. This lets it fly completely under the immune radar. It even shuts down its Krebs cycle, shifting entirely to anaerobic glycolysis. This deep metabolic hibernation is exactly how they manage to survive long-term inside those tissue cysts. Section 5. Morphology of Plasmodium species. Let's leave the tissue cysts behind, jump into the bloodstream, and look at the morphological markers of human malaria. Just like Toxoplasma, Plasmodium relies heavily on surface proteins to breach our cells. Their merozoites use merozoite surface proteins, or MSPs, to grab onto specific red blood cell receptors, like band 3.
Once inside, the parasite physically alters the host cell, and this looks very different depending on the species. Plasmodium vivax will actually enlarge the host red blood cell, leaving behind a fine red stippling known as Schuffner while forming highly irregular amoeboid-shaped trophozoids. Plasmodium falciparum, on the other hand, keeps the host cell its normal size. It produces multiple young ring stages within a single cell, and eventually forms its signature mature gametocytes, which have a distinct banana or crescent shape.
Looking at gem-sustained smears makes these differences immediately obvious. You can see the P. vivax trophozoite spreading out amoeboid style through a visibly swollen, speckled red blood cell. Now contrast that with the P. falciparum smear. The red blood cells there are totally normal in size, and you can clearly spot that defining crescent-shaped gametocyte. That morphological structure is completely exclusive to the falciparum species.
Section 6. Morphology of intestinal coccidia. Let's wrap up our structural analysis here.
Identifying intestinal coccidia oocysts really comes down to an ascending scale of size and internal structure. Cryptosporidium is the smallest of the bunch, at just 2 to 4 micrometers in diameter. Inside, it contains 4 naked sporozoites with absolutely no sporocyst holding them. Stepping up in size, we have Cyclospora cayetanensis at 8 to 10 micrometers. This one develops a dual sporocyst structure, with each sporocyst holding 2 sporozoites. Finally, you have the massive Cystospora belli, which can measure up to 33 micrometers. It also uses a dual sporocyst architecture, but it packs 4 sporozoites into each one. With Cystospora belli, you can actually watch the structural development happen under a microscope. The immature oocyst starts off as just a single, unsegmented granular mass. But as it matures, that internal mass literally divides into 2 distinct sporocysts. This mature dual sporocyst form is the infective stage that essentially defines the biology of the species. So we've just seen how incredibly precise structural components and deep metabolic rewiring allow these parasites to absolutely dominate host cells. But their evolutionary history might actually be their greatest weakness. If the apicoplast is literally just a remnant of a cyanobacteria, one that renders these complex eukaryotic organisms vulnerable to standard, everyday prokaryotic antibiotics, it leaves us with a pretty fascinating question to close out this explainer. How many other unrecognized prokaryotic vulnerabilities are hiding inside these eukaryotic parasites, just waiting to be discovered?